of desired gene is ensured if virus infects some of the germ cells of the animal. The major concern regarding the retroviral method has been the probability of reconstructing infectious virion particles upon transfection. There are reports of the formation of infectious particles, but the problem lies in the packaging extracts that previously harbored retroviral sequences in which packaging signal sequences were absent. But recently, the cell lines used for packaging are transfected with constructs coding for viral particles and transferred to different chromosomal locations. This process invariably removes the threat of generation of infectious virions. The most common retrovirus is used for stable integration of the gene in the genome, or lentivirus and simian immunodeficiency virus. The diseases like severe combined immunodeficiency, cystic fibrosis, or even hemophilia A can be treated with this strategy in the future, as clinical trials are in progress. Figure 12.28 Retrovirus-mediated gene transfer for producing transgenic mice. Mouse embryos devoid of zonal cells are cultured in the presence of myeloid leukemia, momo virus-producing cell lines. The viral genome integrated in the cell lines is altered and contains the desired gene of interest. The modified viruses integrate into the embryo middle genome, and the gene of interest can be found in all cells of resultant offspring bottom. Although the above described techniques do result in transgenic animals, the rate of success of live birth of animals containing transgene is very low, given that genetic manipulation does not lead to abortion. The F1 generation produced is checked for the expression of the transgene as it may be chimeric, depending on the approach adopted for transgene insertion. The germline chimeras are inbred for 10 to 20 generations till homozygous transgenic animals are obtained. The excess embryos can be cryopreserved for future implantation and production of transgenic animals. With the above discussion, the various applications of transgenic animals can be seen in biotechnology, which are summarized below. 1. In basic research, transgenic animals are useful in identifying function-specific genes in complex systems by knocking in or out the expression of specific genes. 2. The transgenic animals are useful in screening toxic compounds. 3. Developmental biology can be studied in a more detailed way by editing genes responsible for certain stages of development. 4. The pharmaceutical companies also employ transgenic animal models for protein antibodies-based drug production as well as screening. 5. The disease models are useful in screening for effective drugs in labs and industry. This method decreases the requirement of a human subject for drug development. 6. The livestock can be genetically modified to increase dairy and meat production. Transgenic animals can also be produced which have more tolerance for climate and diseases. 7. In clinical applications, transgenic animals are developed for the production of tissues and organs for xenografting. Stem cells in livestock. Embryonic stem cells have capacity to differentiate in any cell type in vivo or in vitro. These cells can also differentiate if injected into the blastocyst and give rise to a chimeric animal. Pluripotent stem cells and induced pluripotent cells also have infinite capability. Somatic cell nuclear transfer (SCNT). Somatic cell nuclear transfer (SCNT) is a laboratory technique in which a viable embryo is formed by a nucleated egg cell and a body cell, which is diploid in most vertebrates. This technique is used in both therapeutic and reproductive cloning. The term therapeutic cloning here implies regenerative medicine, in which the controversial ethical use of embryo stem cells was required. This technique can be considered an answer to all the ethical issues related to embryo stem cells. In this technique, the female gametocyte egg cell is collected from a donor with consent by ovarian stimulation. The genetic material from the donor egg cell is removed, resulting in a nucleated cell which has only cytosol. Now, the second cell, which is somatic in origin, can come from any body tissue like skin, liver, and fat, to name a few. This somatic cell containing a nucleus is fused with a nucleated egg cells by electrical pulses. The fused cell is again stimulated by electrical pulses and begins to undergo cell division. It is proposed that factors present in the cytosol of egg cells cause the division and reprogram the nuclei to form a blastocyst, an early embryo stage with about 100 cells. The blastocyst can be then transplanted into a surrogate mother for further development into a full organism. Otherwise, the blastocyst stage can be used for obtaining embryonic stem cells which can be genetically manipulated for stem cell-based therapy or regenerative medicine. Dolly the sheep was the first animal to be cloned by SCNT technique in 1995. The success rate for Dolly was very low as it was the only calf that survived to adulthood out of 277 planted embryos. But now with a better understanding of egg cell factors and technology, in 2018 first primates, 2 female crab-eating macaques were cloned. A general schematic view of the steps involved in the process is shown in figure 12.29. Of self-renewal while retaining the property to differentiate in any cell type. While stem cells are derived from bone marrow, adipose tissue has limited capacity to differentiate in multiple lineages. The S-based approach would have many applications in domestic animals such as pigs, cows, and dogs, which are excellent long-term models for regenerative medicine and other medical research because of their similarity to human physiology as compared to small animals like mice. Moreover, mice models with targeted gene manipulation fail to produce a human phenotype. Amongst livestock, pigs are the most suitable model system as their physiology and organ size are very close to humans. There has been constant effort to derive pluripotent stem cells from pigs and induce them to differentiate along various lineages. There have been attempts to drive the differentiation of porcine pluripotent stem cells to differentiate, but this has been inconsistent and difficult to achieve. Although there are reports of chimeric partially humanized pigs which can be later used for the production of complete human organs, it is also shown that pigs can also be used for producing severe combined immunodeficiency (SCID) phenotype, which is very useful for testing stem cell therapies, whole organ transplants, and other clinical and surgical procedures where immune tolerance is desired. Another application of porcine muscle stem cells has been known for the production of meat for human consumption. This requires techniques for bulk culturing of cells and formulating ideal growth conditions so that cells have the taste and consistency of meat obtained from the animal body. All the above discussed applications of stem cells in livestock have still not been achieved completely and many remain futuristic. But with the progress in stem cell science, these futuristic applications may turn into reality. Figure 12.29 A general schematic view of the SCNT process. The donor egg cells are enucleated and fused with a nucleus isolated from somatic cell. The enucleated cell and nucleus are fused by electrical pulses and the fusion cell is allowed to grow and multiply. The nuclei derived from somatic cell are reprogrammed to form blastocysts after fusion, and then at about 100 cell stage it is transplanted to surrogate mother in case of reproductive cloning. Otherwise, as these cells are embryonic stem cells, they can be genetically modified and used for stem cell-based and regenerative therapies. By SCNT technique, pluripotent stem cells which are genetically the same as the patient cells can be obtained without the destruction of a viable embryo. SCNT is useful in creating cell-based model systems for screening the drug and therapies for many important genetic and physiological diseases like Parkinson's, myopathies, and cardiac diseases. The SCNT can enable researchers to treat the diseases by stem cell-based approach as well as screen the effective drugs through the cell-based experimental models created by this lab technique. But SCNT is a labor-intensive technique with low success rate. The probability of obtaining a viable embryo post-fusion of egg and somatic cell depends on factors such as the cell cycle state of both cells. But enthusiasm for SCNT-based stem cell research has decreased as other methods of generating pluripotent stem cells have been devised like one in 2007 by Yamanaka et al. But still, how iPSCs are similar to ESC is still under investigation until the final answer. SCNT stem cells will be relevant for stem cell-based therapies. Biotechnology and dairy industry. By 2050, it is estimated that milk and meat production requires to be increased by 70 to 80% relative to current production to meet the population demand. Biotechnology related to food production, processing and preservation comes under the area of food biotechnology. The process is designed and streamlined by food technologists help in the prevention of spoilage by unnecessary microorganisms that cause toxicity like Clostridium botulinum and other bacterial species, and degradation of nutritional components of food items. It also helps in identifying the presence of harmful viruses and bacteria in food, pesticides, and other chemical agents which may be harmful for human consumption. All over the world, milk is considered as the most wholesome food offered by nature. Because of its high dietary nutritional value, milk is used for manufacturing a large number of its versions like skimmed, lactose-free, and caseophyllin prevents aggregation of platelets and hence has antithrombotic activity. Probiotics are now closely associated with the dairy industry. The term was first used by Lillian Stillwell in 1965 to describe the substance contained in one microorganism which boosts the growth of another. Currently, probiotics are referred to as living organisms administered in fixed numbers to promote the healthy gut flora. In general, the lactic acid-producing bacteria are sufficient to ensure the healthy gut flora. Besides this, probiotic food supplements are used for the treatment of diarrhea, calming down the symptoms of lactose intolerance, reduction of blood cholesterol, etc. New versions of many food items available in the market like milk, yogurt, and food for infants have added probiotic according to approved guidelines. Functional genomics for livestock production. The term livestock means animals raised in agricultural settings to produce commodities such as food, clothing, and labor. Livestock can range from cattle, sheep, goats, and horses, birds like poultry, fisheries, and even insects like honeybees and silkworms. Although livestock species have been domesticated for thousands of years, they have been improved through selective breeding for better yield and production. But now as the world is continuously expanding in terms of population, more urbanization and increasing incomes have resulted in increased demand for food items such as dairy, meat, and other animal products. The challenge of providing the growing population becomes more difficult as economic factors related to animal well-being and sustainable practices need to be taken care of. With conventional selective breeding, the rate of incorporating desired genetic traits is very slow. To accelerate the process of genetic evolution, biotechnological intervention is required. Molecular genetics aiming to improve the yield and quality of animal products as well as the good health and well-being of animals are indispensable. 3 innovative techniques. Whole milk. It is also used for making fermented dairy products like cheese, yogurt, kefir, and bulgur. Butter, milk whey, ice creams, and some enzymatically modified milk products are commodities that are obtained from milk. Recent developments in biotechnology in particular can enormously benefit the dairy industry by not only increasing the overall quality and safety of processed dairy food but also by making the commercial values more affordable for local consumption and export. Since the ultimate purpose of the dairy industry is to provide high-quality, nutritious, healthy, and inexpensive food to consumers, so biotechnological interventions were necessary at different stages of milk production. Besides being nutritional food, milk contains bioactive components such as immunoglobulins, hormones, growth factors, cytokines, nucleotides, enzymes, polyamines, and peptides. The bioactive peptides present in milk are activated by gastrointestinal enzymes while being digested or during food processing. The commercial production and applications of these peptides depends upon the production and their innovative formulations for consumption. The therapeutic compounds from dairy products also need to be clinically verified for their pharmacological action and their health effects. Although all the contents of dairy products are not characterized, there are reports which report the antihypertensive activity, immune modulatory properties, and antimicrobial activity against the pathogenic microbes. The proteins which come from whey like lactoferrin, beta-globulins, and alpha-lactalbumin have immune modulatory functions. Also, the probiotic lactic acid bacteria are involved in generating immune regulatory peptides via enzymatic degradation of milk proteins. Other peptide derivatives from milk proteins have morphine-like activity and are called seasonomorphins. These opioid agonists prolong the gastrointestinal time, thus helping in regulating diarrhea and stimulating somatostatin and insulin secretion. While opioid antagonists such as casoxins affect the release of endogenous pain-reducing endorphins, used in general, however, the technique is not restricted to tissue culture but also includes cell, organ, and suspension culture. It has huge importance in the field of plant biotechnology and has especially revolutionized crop improvement programs. It is the foundation of many aspects of plant biotechnology. The results obtained in vitro are rapidly transferred to the fields. Molecular biologists have also been intrigued by the field of plant tissue culture. The field has attracted the attention of industrialists and plant breeders due to its economic importance. This field also deals with investigating the factors responsible in the patterns of growth, differentiation, morphogenesis, and metabolism of plants. The earliest attempt at plant tissue culture was done in 1756 by Henri Louis de Maldumonceau, who detected the formation of callus during the healing of a wound in the plant. Cell theory was postulated by Schleiden and Schwann after extensively carrying out microscopic studies. This theory suggested that cells were the fundamental unit of life and were capable of autonomously replicating. In 1902, a German botanist, G. Haberland, successfully cultured fully differentiated plant cells which were extracted from various plants. This marked the beginning of plant tissue culture. Cell doctrine made further contributions to this field by revealing that plant cells were totipotent. Further developments took place in this field when various chemicals such as auxin, cytokinin, vitamins, and other hormones were identified and how they affected the differentiation and growth of plants. Some of the important contributions in this field were made by Novaport, White, and Gotheret. The in vitro culturing techniques were enhanced after the discovery of plant growth regulators and their effects on the multiplication of cells. Cultures were tailored to the needs of the explants. Tissues, cells, embryos, root tips, anthers, and protoplasts were all grown in a suitable culture medium to maximize the growth. Are fundamental for accelerating the genetic improvements in livestock and other animals of commercial importance, and they are as follows. 1. Reproductive technologies like artificial insemination, which is a low-cost and technically less demanding practice to improve the genetic traits in livestock. 2. Molecular biology technologies like SCNT and chimeric animals through modified stem cells. But these techniques require a lot of experienced personnel and finances. 3. Genotyping tools which allow for gene mapping and incorporating that information in breeding programs for marker-assisted selection techniques. As more and more genome sequences of various animals like cows, pigs, and chickens, etc., are available, the sequences are curated and analyzed. The traits linked to single nucleotide polymorphism are being analyzed, and panels for high-density SNPs are available for chickens, pigs, and cattle for whole genome studies for important traits. Moreover, many biomedical research projects have used livestock animals for achieving goals in the area of comparative genomics and physiology, cloning, reproductive cloning, and production of pharmaceuticals. The comparative physiology, long generation time, and availability make them an ideal model system for biomedical research. For example, cows are universe animals and hence form good model system for reproductive research, while pigs have been used as a model system for studying diabetes and obesity. Apart from this, baby pigs are a model system for studying child nutrition and evaluation of baby food and supplements. Plant biotechnology. Plant tissue culture. Overview of tissue culture. Plant tissue culture is a technique that allows culturing of the explants in vitro under aseptic and extended conditions in a nutrient medium of known composition. The term tissue culturist G. Haberland is known as the father of tissue culture since he was the first to initiate the concept of in vitro culturing of plant cells. The contributions of other scientists in this field are indicated in table 12.6. Braun, in 1959, was the first scientist to generate an entire plant from a mature plant cell. In India, the tissue culture was led forward by Kim Hashwari, who worked on in vitro culturing of plants and discovered the haploid production. Name of scientist, year, contribution. Haberland 1902 first to endeavor in vitro culturing of plant cell, Hennig 1904 culturing the embryogenic tissue of crucifers, Robbins 1922 culturing of roots, in vitro Lagac 1925 zygotic embryo culture, and Lindenwhite 1934 culturing of roots of tomato, Novaport, White and Gotheret 1939 successfully established indefinite callus culture, Braun 1941 culturing of crown gall tissues, Lou 1945 culturing from the tip of the stem. Miller 1955 discovered kinetin. Miller and Scoop 1957 discovered organ formation is regulated by auxin. Types of plant tissue culture. Seed culture. In vitro culturing of seeds can be carried out to produce plants or seedlings. Seed culture is an unmatched way to raise a sterile seedling. In order to maintain the aseptic condition, seed culture can be carried out to generate various explants which are aseptic. This helps in better maintenance of the sterile conditions needed for experimentation. Embryo culture. In vitro culturing of an immature or mature embryo under aseptic conditions to produce a plant is known as embryo culture. Seed intergeneric hybrids and interspecific hybrid productions are also possible from embryo culture. Occasionally, if hybridization is attempted, the embryos from different species fail to mature because of embryo endosperm incompatibility. This problem is solved by employing embryo culture techniques. Dormancy is seen in certain plants. This can be caused due to mechanical resistance or chemical inhibitors acting on the structures protecting the embryo. In such cases, the embryo remains viable and hence can be isolated. These embryos grow to form a viable plant in a suitable nutrient medium. Figure 12.30. Figure 12.30 procedure for embryo culture. In vitro culturing of an explant in a nutrient media is carried out as shown in the diagram. Root culture. In vitro culturing of radical tips excised from seeds germinated aseptically under regulated conditions is known as root culture. Root tips arising from seedlings are highly sensitive to the chemicals used in surface sterilization. Plants growing in vivo cannot be used for the extraction of root tips because their roots are buried deep in the soil. To overcome this problem, root tips are extracted from aseptically growing germinating seeds and the culturing is carried out. Figure 12.31. Root culture is usually carried out in a liquid moving media. The root tips are cultured in a way that they exactly resemble the roots of their counterparts grown in vivo. The lateral tips of roots of the main roots, if cut and transferred from a single root culture into a fresh medium at regular intervals, would help in the successful cloning of the excised roots. The growth of the cultured roots is measured by calculating the dry and fresh weight, the total length of laterals per culture, the number of emergent laterals, and the increase in length of the main axis. Another variant of the root culture is the hairy root culture. It is also known as transformed root culture. It provides insight into the metabolic pathways in the plant and is extremely helpful in deriving secondary metabolites. Often, plant genetic engineering is employed in hairy root culture is used to obtain recombinant proteins. Are genetically identical and hence can be used for examination purposes. 2. Nutritional requirements for plants can be studied by continuously culturing isolated roots. The results can be used to efficiently grow the plants. It was found that if some plants were supplemented by thiamine, the plants grow indefinitely. The nutritional requirements for tomato roots were also found. 3. The formation of bud and lateral roots, nodulation, and cameal activity of plants can also be monitored. 4. Root culture provides an ideal platform to study the effect of the shoot on the growth of roots. 5. Commercial production of secondary metabolites can be done with the help of the root culture technique. 6. In some cases, the cultured root tip did not give rise to secondary vascular tissues. This was an indication that certain substances from a shoot affect the development and formation of cambium in roots. Hence, root culture could be deployed to study the controlling factors in the development of secondary vascular tissues in plants. Callus culture. In 1893, Rietzschner first observed the formation of callus in the isolated stem segments of populus. Callus formation is typically seen when the wound of a plant begins to heal. It is formed by repeated division of the parent tissue. Callus is an unorganized mass of amorphous and parenchymates tissue. In vitro culturing of these unorganized and indifferentiated mass of cells in aseptic conditions and in proper nutrient media is known as callus culture. The procedure for callus culture is shown in figure 12.32. Importance of callus culture. 1. A phenomenon known as morphogenesis or organogenesis allows the regeneration of an entire plant from a callus tissue, in large numbers, by regulating the hormonal and nutrient constituents in the media. A process called somatic embryogenesis. Rhizobium rhizogenes is a naturally occurring soil bacterium that contains root plasmids. These root-inducing plasmids can infect the roots of the plants. The roots then become a source of food for these bacteria. These bacteria also cause the roots to grow abnormally and produce opines. These abnormally grown roots do not need hormones and hence are particularly easy to culture in a media. They are capable of growing indefinitely and are neoplastic. These neoplastic roots have a greater biochemical and genetic stability and a higher growth rate. Upscaling development of appropriate vessels or bioreactors to culture the sensitive and delicate hairy roots are the biggest constraints in employing the hairy root culture technique commercially. Figure 12.31 root culture for tomato plants. In vitro culturing of an explant in a nutrient media to facilitate the growth of roots is carried out as shown in the diagram. Importance of root culture. 1. This culture technique is useful in extensively studying the metabolic pathways in plants. The root culture enables us to obtain exact copies of isolated roots or clones. These 4. It is easier to perform biochemical analysis on callus culture. 5. It is beneficial in acquiring the secondary metabolites which are commercially very important. Secondary metabolites can be obtained without the need to necessarily kill the entire plant if the tissue from a medicinal plant is grown in vitro. This provides an alternative technique and aids in the conservation of plants. Would generate a cluster of embryos from the somatic cells of the callus. This is also achieved by manipulating the nutrient and hormonal constituents. Whole plants can be obtained from somatic embryos. 2. Karyotypic or genetic variability can be easily detected in callus tissues. Hence, they serve as a good source to obtain genetically variable plants from the callus tissue. 3. It becomes effortless to produce a cell suspension culture in a moving liquid medium from callus culture. Figure 12.32 Callus culture for tomato plants. In vitro culturing of an explant in a nutrient media to facilitate the growth of callus is carried out as shown in the figure. Organ culture. In vitro culturing and maintenance of an organ or part of an organ in a manner which permits the preservation and differentiation of the function and architecture is known as embryo culture. In 1922, W. Cote and W. J. Robbins were the first ones to culture the excised root tips derived from a wheat seedling grown which was grown aseptically. In the year 1930, White successfully cultured the root tips of a tomato seedling which was grown aseptically. Importance of organ culture. Organ culture opens up a novel dimension in the developments in horticulture and agriculture. The growth factors and nutrients supplied by other plant body parts to the organ and also from the external environment can be monitored easily. Hence, this serves as an ideal experimental setup for studying them. The interdependence of growth factors, hormones, and organs can be studied in this culture technique. Figure 12.33. The organs can be used to study the precise problems in morphogenesis and also for examining the sites for the biosynthesis of growth compounds and specific metabolites. Figure 12.33 Organ culture and its branches. The various cultures that can be carried out from the organ culture. Protoplast culture. A plant cell without the cell wall is known as protoplast. In vitro culturing of isolated protoplasts is done in a semicellular liquid medium in petri dishes. The components of the medium are same for protoplast culture as they are for suspension or callus culture. However, it is advised to keep the calcium concentration high to maintain the membrane's integrity. The medium is also supplemented with a higher amount of sugar. Growth regulators and vitamins are used for the requirements of differentiation, callus formation, and cell division. Another important criterion is to keep the plating density high. The usual plating density for protoplast culture is 1 × 10⁴ to 1 × 10⁵ protoplasts per milliliter. Higher density ensures the viability of the protoplast and the density is subsequently reduced in the later stages. Stages of protoplast culture. There are almost 4 different stages of culturing protoplasts. The protoplast prepares itself for cell division by regenerating the cell wall around itself. Successive cell divisions result in the formation of callus. Embryogenetic or organogenetic differentiation in the callus leads to the formation of embryo or plantlets. All the plantlets or embryos generated possess the same genetic characteristics as they develop from one single protoplast. Figure 12.34. Figure 12.34 Different stages of protoplast culture. The protoplast forms a cell wall and it undergoes successive divisions and form a callus. Medium and genotype of the donor plant as well as other physical factors. After the formation of callus, subculturing is performed with different combinations of hormones at regular intervals for additional differentiation. Figure 12.35. The callus then develops to form an embryo or plantlet. After the formation of an embryo or plantlet, not all cells undergo cell division. Cell division is affected by several factors such as hormones. Figure 12.35 The different stages of protoplast culture. Protoplast isolation and culture and its regeneration are depicted in the figure. This method is preferred. The obvious con of this technique is that it is extremely difficult to isolate single colonies derived from a parent cell. Agar culture. The protoplast culture medium is almost always solidified using agarose. The amount of protoplast used here is double of what is used in liquid culture. The suspension is mixed with agar and plated in a petri dish. This immobilizes the protoplast and keeps them in the same position. The medium can only be changed in this culture technique after the callus formation occurs. Droplet culture. The protoplasts are suspended in a liquid culture medium and are positioned on the petri dish in the shape of a droplet. Protoplasts tend to cluster in the center of the droplet. In this method, the media can be changed at regular intervals. Co-culture. In this technique, recently isolated protoplasts are mixed with the already grown protoplast suspension and the mixture is plated. This practice induces division. Growth factors aid in inducing the growth and development of the newly isolated protoplasts. Hanging droplet culture. In this technique, a thin layer of liquid media is added to the petri dish to ensure that the inside environment remains humid. The protoplast suspension is placed on the inner surface of the lid of the petri dish in the form of an inverted droplet. The con of this technique is that a very small number of the protoplast forms a cell wall and they undergo successive divisions and form a callus. Medium and genotype of the donor plant as well as other physical factors. After the formation of callus, subculturing is performed with different combinations of hormones at regular intervals for additional differentiation. Figure 12.35. The callus then develops to form an embryo or plantlet. After the formation of an embryo or plantlet, not all cells undergo cell division. Cell division is affected by several factors such as hormones. Figure 12.35 The different stages of protoplast culture. Protoplast isolation and culture and its regeneration are depicted in the figure. This method is preferred. The obvious con of this technique is that it is extremely difficult to isolate single colonies derived from a parent cell. Agar culture. The protoplast culture medium is almost always solidified using agarose. The amount of protoplast used here is double of what is used in liquid culture. The suspension is mixed with agar and plated in a petri dish. This immobilizes the protoplast and keeps them in the same position. The medium can only be changed in this culture technique after the callus formation occurs. Droplet culture. The protoplasts are suspended in a liquid culture medium and are positioned on the petri dish in the shape of a droplet. Protoplasts tend to cluster in the center of the droplet. In this method, the media can be changed at regular intervals. Co-culture. In this technique, recently isolated protoplasts are mixed with the already grown protoplast suspension and the mixture is plated. This practice induces division. Growth factors aid in inducing the growth and development of the newly isolated protoplasts. Hanging droplet culture. In this technique, a thin layer of liquid media is added to the petri dish to ensure that the inside environment remains humid. The protoplast suspension is placed on the inner surface of the lid of the petri dish in the form of an inverted droplet. The con of this technique is that a very small number of protoplasts can be cultured in this way. Bead culture. Polymers such as keratin or alginate are mixed with protoplast suspension and shaped in the form of a bead by dripping the mixture in the liquid media. The mixture is slowly shaken and the protoplast is cultured in it. Methods of protoplast culture. Various methods are used for culturing of protoplasts. These include feeder layer technique, immobilized bead culture, hanging droplet culture, co-culture, droplet culture, agar culture, and liquid culture. Figure 12.36. Figure 12.36 Different cultural techniques for protoplast culture. The figure depicts various methods employed in protoplast culture. Figure A shows a liquid medium culture. Figure B depicts a feeder layer culture. Figure C is a representation of microchamber culture. Figure D shows the hanging drop culture method used for protoplast culture. Liquid culture. This method has an edge over the other methods as it permits effortless solution and transfer. Protoplasts are capable of faster division in liquid medium. It also allows for regulation of the osmotic pressure of the medium which can be efficiently reduced during the successive growth of protoplasts. During the early developmental stages of protoplasts, to carry out micropropagation, sterilization of tissues and practices need to be done to ensure that there are no other bacterial or viral contaminations into the clone that we are trying to make. We also need various forms such as auxins which promote callus formation. Upon growth of callus, it can be cut and transferred to another media with cytokinin and gibberellin. Cytokinin induces the root and shoots growth and gibberellins are sometimes needed to induce the growth of shoots. After the growth of plants, they are removed from the media and transferred into the soil where they can be grown naturally. Axillary bud approach. Axillary bud propagation is one of the methods used for micropropagation in plants. It is a very widely used approach and is also regarded as extremely effective for obtaining regenerated plants with guaranteed genetic stability. A dormant axillary bud is induced to grow in vitro in a medium containing plant growth hormones. To activate the dormant axillary bud and induce growth in it, cytokinins are used. After the bud has come out of the dormant phase, it is grown in a media supplied with cytokinin and auxins which promotes shoot and root growth. Most of the plants such as rice, barley, almond, turmeric, tea, apple, etc. are grown using this technique. This technique is very simple and reliable for clonal propagation. It is adopted for micropropagating plants on a larger scale. Lateral meristems cause plants growth through the normal ontogenetic route which is exploited for micropropagation of plants in the culture. The axillary buds are stimulated to form a shoot and in this technique, bud, shoot tip, and meristem culture are carried out. Figure 12.37. Cells do not undergo the differentiation and the growth and development of shoots occur from the meristematic tissues which were already existent. Hence, in this method, we are able to maintain genetic stability as no possibility for variation occurs. The progenies resemble the experimental sample and are also epigenetically stable. Feeder layer. The protoplasts are very efficient in enhancing the growth of other protoplasts. This knowledge is exploited and a feeder layer is designed. This feeder layer consists of protoplasts which have been irradiated with X-rays. These protoplasts are living but they do not divide. Hence, it serves as an ideal environment for protoplast culture by providing all the necessary growth requirements. Micropropagation. It is another method used to clone a plant. Micropropagation is being used by many people for cloning purposes. Most of the cells in a plant are totipotent cells. A lot of tissues in plants are stem cell type tissue or meristematic tissue. For example, on the top of the plant is found apical meristem. These apical meristems are constantly dividing cells. Tissues are derived from the shoot apex of the plant and are aseptically cultured. The media is supplied with a plant growth hormone auxin. This gives rise to callus. The callus can be transferred to an agar gel with another type of hormone known as cytokinin. Another hormone known as gibberellin is also added to the media. Gibberellins help in the promotion of shoot growth. It also helps prevent the plants to go into the state of dormancy. The miniature plant that now we have is identical to the host plant. This can now be transferred into the soil and will have a replica of the original host plant, and we have carried out the process of cloning successfully. If the different cells are extracted at different stages, then very quickly just by removing some tissues or some cells that have the potential to grow and have the same genetic makeup as the original plant, we can prepare additional clones. This technique is called micropropagation. Micro means only a tiny amount of cells are needed to start the process, and propagation means to propagate or make more. This technique is extremely effortless to be carried out on a small or large scale using very minimal equipment. Commercially, it acts as a hindrance in the conservation of biodiversity for plants. When the plants are cultured in vitro, they undergo repeated differentiation and redifferentiation which exposes them to dramatic stress. Frequently, the genetic and epigenetic changes in plants or plant organs are caused because of this dramatic stress. Other forms of tissues are more susceptible to genetic changes than the meristematic tissues. Hence, axillary bud culture is considered to be the technique that minimizes the risk of genetic instability. Sometimes, no substantial genetic variation is seen in axillary bud culture. Molecular markers such as retrotransposon microsatellite amplified polymorphism, RFLP, RAPD, ISSR, and cytophotometry were used to map the genetic differences between the parents and progeny from the axillary bud culture. Culturing of axillary bud and somoclonal variations. Somoclonal variations are one of the major problems associated with the axillary bud culture technique. It involves retrotransposons and transposons activation, gene activation and silencing, changes in the gene sequence, phenotypic mutations, and cytological abnormalities. Hence, it can be regarded that somoclonal variations occur genetically as well as epigenetically. Culturing of axillary buds which results in somoclonal variation serves as a treasured source of variations occurring genetically for improving the crop by selecting the new variants. These novel variants might display higher yield, improved quality, or increased resistance to diseases. It could also result in off-types which reduces the value of plants. Figure 12.37 schematic representation of axillary bud culture technique. Organogenesis. During culturing, an X plant is supplied with an appropriate concentration of growth hormones such as auxins or a mixture of auxin and cytokinin. Supplementing a medium with growth hormones ensures that the X plant would generate the formation of callus. Media containing growth hormones to induce callus formation is termed initiation media or callus-inducing media. For a sustained amount of time, the proliferation of the massive callus is carried out. If subcultured in the same medium for a longer duration, callus proliferation continues for a long time. For organogenesis, the main purpose is the regeneration of a plant organ or a plant from the callus tissues. Cellular totipotency existing in all the cells of a callus is the chief reason why regeneration can take place. However, callus grown in a controlled medium shows reduced totipotency. This potential of callus to form totipotent cells can be enhanced by the regulation of hormones and nutrients in the media. Adjusting the relative levels of cytokinins and auxins initiate the type of organ primordial formation in the callus culture. This was first demonstrated by the scientist Skoog and his coworkers. The development of meristematic centers, which were the areas of activity during the development. Some errors might occur in the developmental programming of organogenesis, which gives rise to an abnormal structure known as organoids. Organoids are generated from the periphery of the callus tissues and are not a result of differentiation in the meristematic cells. Hence, even though they have ground, vascular, and dermal tissues, they are different from the true organs present in a plant. In the meristematic cells, there are present meristemoids, which are localized group of cells. These meristematic cells give rise to callus, and their further differentiation produces roots and/or shoots. Process of organogenesis. An X plant such as a tissue derived from plants, cells extracted, isolated protoplasts, and microspores can be cultured in vitro. The X plant can be grown in an appropriate medium which induces proliferation in the cells and results in the formation of callus. This callus then differentiates into a root or shoot or both. Figure 12.38. The growth of the root or shoot is dependent on the nutrients present in the medium. Trial and error method is used to standardize the medium. Irrespective of the nutrients and hormones provided, the callus might remain in an undifferentiated stage. This unlimited proliferation is stopped once the organ formation begins. The cells of the callus grow to form actively dividing meristematic cells, which then give rise to root or shoot primordia. Rhizogenesis or budding of adventitious shoots follows callus growth. The capacity to form roots or shoot bud decreases with the increasing subculturing of the callus. Rhizogenesis occurs more often than other forms of organogenesis. Shoot formation becomes increasingly difficult to induce after the formation of roots. However, in cases where shoot formation precedes root formation, the formation of roots occurs at a later stage. Root and shoot formation can also occur simultaneously depending upon the composition of growth hormones in the medium. A general characteristic of organogenesis is the formation. Dividing cells was a result of differentiation. Further differentiation of these meristematic cells resulted in the generation of shoot and root primordia. When the ratio of auxin, cytokinin was kept high, the formation of roots was seen, and when the ratio of auxin, cytokinin was kept low, the formation of shoots was seen. A moderate level of auxin and cytokinin resulted in the development of callus tissues. This was observed by Skoog and Miller in the year 1957. Since the callus cells are totipotent, this property can be exploited for the formation of either shoot or root or both. Organogenesis is a term used to define the production of adventitious shoots or roots from the callus culture in the culture medium. History. In the year 1944, F. Skoog first indicated that in vitro organogenesis could be controlled by the help of chemicals to a certain extent. Skoog also discovered that auxin stimulated the formation of roots when added to the nutrient medium. Auxin inhibited the formation of shoots. However, in the presence of inorganic phosphate or sucrose, the effect of auxin on shoot initiation inhibition was reduced. In 1948, F. Skoog and C. Sui discovered that auxin's effect could be chemically reversed by adding sulfate. Miller and Skoog in the year 1957 discovered that auxin and cytokinin had a controlling effect in the induction of callus, root or shoot formation in a cell culture. Types of organogenesis. The development of primordial or adventitious organs from an indifferentiated mass of cells in a tissue culture set up by differentiation of the indifferentiated cells is known as organogenesis. The type of organogenesis in which the callus tissues differentiate to produce adventitious roots is known as rhizogenesis. A type of organogenesis in which the callus tissues are differentiated to produce adventitious shoot buds is known as collagenesis. Zygote develops into an embryo. This process is known as embryogenesis, which happens within the seed. The endosperm seed coat and embryo forms the primary parts of the seed. The embryo consists of all the cells and tissues which are required for the development of a plant body. The endosperm serves as a source of stored food which provides the nutrients for the developing embryo. The seed coat forms a protective layer that encases the seed. In many cases, the embryo after being formed stops growing and enters into a state of dormancy. During favorable conditions, the embryo resumes rapid growth and development and matures into a plant. The embryo can be divided into two main regions: the hypocotyl which develops into the root system and epicotyle which forms the shoot system. The young juvenile leaves near epicotyle are called cotyledons. These cotyledons provide stored food in the form of starch and proteins to the germinating embryo. The name suggests the embryo of a monocot plant has one cotyledon, whereas the embryo of a dicot plant has two cotyledons. The monocot plant stores energy in the embryo, whereas the dicot stores energy in the cotyledons. The seed coat also protects the embryo from drying and from any other forms of physical injury. The point of attachment of seed to the seed wall is called hilum. The small opening in the seed coat near the hilum is called the micropyle. In monocots such as grass, the single mesh of cotyledon is termed as the scutellum. The plumule and radicle are enclosed by protective structures known as coleoptile and coleorhizae respectively. The fertilized egg called the zygote is unicellular in nature. This zygote through a series of events gives rise to a multicellular embryo with well-differentiated organs which embody within itself the multivariate properties of an adult plant. The series of events that leads to the formation of a mature embryo from a unicellular zygote is termed as embryogenesis. Embryogenesis can be divided into 5 distinct phases. The two-celled stage, globular stage. Of shoots followed by the appearance of tiny hair-like root structures. Callus develops a pale green or green pigmentation when the formation of shoots occurs. It could develop a yellow coloration during rhizogenesis or might not change its coloration at all. The prolonged culturing of callus reduces its ability to proliferate. This might be attributed to the physiological or genetic changes occurring during the repeated culturing or because of the composition of the nutrient media. The genetic variations might cause cryptic chromosomal rearrangements, polyploidy, or aneuploidy. Such alterations result in loss of totipotency in the callus. The physiological changes that might occur are loss of morphogens or endogenous factors present at important stages of growth. Cells' inability to synthesize such compounds results in the diminished organogenetic potential of the callus tissues. A lot of studies have been carried out to investigate the effect of phytohormones on organogenesis. It was Skoog and Miller who in 1957 proposed the concept of appropriate balance of auxins and cytokinins for organogenesis. Embryogenesis. Embryogenesis is a common term used to define the formation of zygote and endosperm. There are various stages of embryogenesis in dicot and monocot plants. In angiosperms, the female gametophyte is also termed as an embryo sac. The ovule contains this female gametophyte. The embryo sac has a polar organization along its micropolar chalazole axis. The chalazole pole has antipodal cells which are present at the opposite ends of the embryo sac. The micropolar pole of the ovule contains the synergids and egg cells. In the center of the cell, there are present two polar nuclei. The pollen tube penetrates the ovule during pollination through the micropyle. The pollen tube delivers the sperm nucleus which fuses with the hap. This fusion results in the production of a zygote. This zygote further develops to form an embryo. The second sperm nucleus fuses with the two polar nuclei present in the center. This produces an extra embryonic triploid endosperm nucleus. After fertilization, the which are the protoderms. This protoderm is the precursor of the epidermis. The delineation of the protoderm establishes the globular stage of embryo which increases in size and cell number by cell divisions of the protoderm and the interior cells. In some plants, the basal cell of the two-cell embryo undergoes a series of transverse divisions resulting in the formation of a hypophysis or the suspensor. The hypophysis is the precursor to the root cortex initial in the central region of the root cap. The suspensor is an ephemeral embryonic structure and it comprises a single row of 6 to 12 cells. There might be a number of variations in the number of suspensor cells according to the plant species. The suspensor cells are responsible for pushing the embryo into the endosperm and also transfer the nutrients and growth factors from the maternal tissue to the embryo during embryogenesis. The suspensor cells usually reach the maximum number of cells in the early globular stage of development and begin to senescence during the torpedo stage. A dramatic transformation of embryo morphology occurs during the transition from the global stage to the heart-shaped torpedo stage. The cell divisions parallel to the embryo surface occur at specific regions of the lateral regions of the globalized age embryo resulting in the emergence of two lobes of cotyledons. The shift in the embryonic symmetry from radial at the globular stage to bilateral at the heart stage represents the initial delineation of the two major embryonic organs, the cotyledons and the axes. The extent to which the shoot apical meristem is activated and tissues are differentiated at this stage varies in different species. Heart stage. Torpedo stage. Mature stage prior to embryogenesis, the zygote might undergo a period of dormancy which brings about many alterations in the zygote. During this period, the vacuole size increases which lead to the accumulation of more cytoplasm towards the chalazole end where usually the first division of the zygote should take place. The nucleus surrounded by a large number of plastids and mitochondria is now pushed towards chalazole end or the apical pole of the zygote. Whereas the micropolar end of the zygote or the basal pole is occupied by one or more vacuoles and a few cell organelles. Besides, it has also been observed that there is an increase in the number of active dictyosomes responsible for wall synthesis around the zygote. The zygote now forms a very highly polarized isolated cell. This zygote also has no plasmodesmotic connection with the remaining ovule. Asymmetric cell division marks the start of embryogenesis. Zygote divides into smaller cells which contain the apical cell and the basal cells. This is the first asymmetric division that provides polarity to the embryo. In certain cases, the first division of the zygote may be vertical, for example in Lauren's or it can be oblique in case of triticum. The growing embryo is known as a proembryo from this stage until it forms the embryo. From this two-cell stage it might form a 4-cell linear or a 4-cell T-shaped structure of proembryo. This 4-cell proembryo divides into an octant. It is in the extent stage that the destiny of the embryo is determined. The octant stage embryo by paraclinal division sets the first histologically detectable tissues. Figure 12.38 Organogenesis and somatic embryogenesis. The roots of embryogenesis and organogenesis are shown in the figure. Advantages of micropropagation. There are a lot of advantages of micropropagation. This can be done quickly and clones can be obtained in a very short amount of time. Even if the rest of the plant is contaminated with any kind of viruses that cause diseases, the apical meristem is free from viruses of any kind. So, when the plants are closed from the apical meristems, they are free from any viruses. Also, if there are plants that are rare, they can be reproduced. It reduces the cost of cloning such rare plants and they do not need to be taken from the wild. Gene isolation. Map-based cloning. Principles of positional cloning or map-based cloning. A molecular marker present in close proximity to the gene of interest needs to be identified. Initial screenings are carried out on a smaller population, 60-150 individuals. The region surrounding the original molecular marker is saturated with various other markers. One of these markers should have a rare possibility to recombine with the desired genes. The population could be increased to 300 to 600 individuals at this point in time. The genomic library is screened for the marker in an attempt to separate the clones which have hybridized with the molecular marker. The sequence gets linked with the probe and then chromosome walking or jumping can be performed to map out the gene sequence. It is carried out using novel markers and screening the population with the marker sequences. The population is usually very large, 1,000 3,000 individuals. The aim is to detect those markers which co-segregate with the gene of interest. Co-segregation is a term used when the marker associated with the allele of the desired gene is expressed when the other allele of the gene is expressed. Co-segregation is extremely vital for the mapping of genes. Hence, this process is repeated unless the markers co-segregate with the desired gene. It is suggested to use a marker closely linked to the gene to a suitable BAC or YAC clone with which the probe hybridizes is found. 3. Creating novel markers from the clones and determining whether or not they are co-segregating with the genes. 4. Co-segregating markers are searched by the repeated screening of the genomic library until they are found. 5. Genes are identified from the BAC or YAC clone after co-segregation of markers. 6. Genetic transformation is performed to liberate the wild-type phenotype. 7. Genes are then sequenced and their functions are determined. In absence of a transformation system, determination of successful cloning becomes increasingly difficult. Recombinational or mutational analysis techniques are more powerful techniques that can be deployed in such cases. Speed up the process and avoid additional screenings. Since the genes are flanked by two markers, it becomes mandatory for the gene to be present in between them. The DNA fragments flanked by markers are introduced into a plant by genetic engineering and the plants are transformed. We know that the desired gene has entered a plant successfully when the transgenic plant shows phenotypic characteristics of the wild-type plant. The open reading frame or encodes the product of the gene. Mostly, only one morph is observed. However, sometimes this is not the case and numerous ORFs are present. An in-depth biochemical and molecular analysis can be performed on these ORFs after they show the presence of genes. The process of positional cloning is carried out as follows. Figure 12.39. 1. A marker linked tightly with the gene of interest in the population to be mapped needs to be identified. Figure 12.39 Principle of map-based or positional cloning. The figure represents the steps involved in positional cloning. Chromosome walking. Chromosome walking is another technique that can be used for the screening of libraries. There are a lot of advantages associated with random DNA fragmentation. If we need to map a larger chromosome, we use the method of chromosome walking to map the gene sequence. A probe is introduced to identify the gene sequence in the genomic library. This probe binds with the gene sequence and also with the number of clones of that gene. During, for instance, if we have a very large fragment of DNA to study that contains a number of genes, this fragment is digested with appropriate endonucleases such as E. coli. The fragments of DNA are then cloned in the bacteriophage lambda. This helps us to obtain a probe which then helps us to detect the next DNA fragment. The next fragment of DNA is cloned in another bacteriophage lambda that helps us to detect the third DNA fragment. In this way, the entire chromosome is mapped. Figure 12.40. In the case of the human genome, the starting point may be a fragment that is known as restriction fragment link polymorphism that might be closely related to various disease loci. A probe is applied in identifying the sequence of a gene from a genomic library. The probe hybridizes with multiple clones and all of them carry with them some part of the fragmented gene. Since the site of cleavage is different in the DNA, the fragments obtained have overlapping sequences. These overlapping sequences are used in the construction of the original genome sequence. Large chromosome regions are reconstructed and characterized in this way. The preparation of a genomic library. The genes are fragmented and each of the probes carries some part of the gene. If the genomic DNA is digested randomly, overlapped DNA fragments will be produced. These overlapping DNA fragments can be used to detect the neighboring DNA fragments of interest. The overlapping of DNA fragments makes it easy to detect the neighboring DNA fragment and this is termed as chromosome mapping. In addition to that, chromosome walking is also used to detect genes whose expression or function is not known but its genetic location is known and this technique is called positional walking. For chromosome walking, we need to have random DNA fragments to produce the genomic library. One of the fragments is used as a starting point or used as a probe so that the next contiguous DNA fragment can be detected. The reason why the neighboring DNA fragment is detected is because the ends of the fragments overlap with each other. In this way, one can move along the whole length of the chromosome and the technique is called chromosome walking. Figure 12.40 Chromosome walking. The average distance between two markers could be lesser than the average length of the insert of the clone library that contains the gene of interest. In chromosome walking, it takes a lot of time and is tedious to find the markers for DNA that is in close proximity to the desired gene fragment. This assumption has been invalidated by the recent advances in technological developments. The advances have completely changed the face of the mapping paradigm. DNA markers are first separated from the gene of interest. This marker is of a lesser size than the average size of an insert in the genomic library. The clone which contains the gene is screened in the library and isolated along with the DNA marker. Chromosome walking and the hassles associated with it can be avoided. It is an entirely different technique and because of the benefits associated with chromosome landing, it is soon expected to eliminate all the other strategies of map-based cloning. It can be used to map genes contributing to the quantitative traits in a plant variety. Molecular markers. Molecular markers are specific genes which have inheritable characters. The identification and characterization of these genes are easy. These genes have some phenotypic characters or code for some specific protein, color, or enzymes. Molecular markers are important for genotype profiling and can be of two types: biochemical markers and molecular markers or genotypic markers. Genotypic markers are genes present on different loci of the chromosomes. In conventional plant breeding, the markers are usually phenotypic. Determining the sequence in which nucleotides exist becomes paramount, hence we use DNA sequencing techniques to identify and characterize the DNA of interest. Conventional sequencing techniques, Sanger sequencing or di-terminator techniques were used to sequence the DNA. Di-terminator sequencing technique coupled with high-throughput DNA sequence analyzer is widely being used for sequencing the DNA. The chains. The figure shows the technique of walking through successive hybridization between chromosome overlapping genomic clones. Procedure for chromosome walking. 1. A clone is selected from the genomic library and one end of this clone is subcloned. 2. Overlapping regions of the chromosome hybridizes the subclone with the second clone. 3. Overlapping regions of the second clone hybridizes with the third subclone, and in this manner, the procedure continues. 4. The overlapping areas are mapped in a restriction map is generated which aids in the identification of novel overlapping restriction sites along the chromosome segment. Chromosome jumping. Another variation of chromosome walking is chromosome jumping. Here we need to construct the jumping libraries. Jumping libraries are constructed in situations where the DNA is extremely large, 50 to 200 kbp. In this case, when the DNA fragment is very large, the gene of interest on that fragment may be far apart. For the construction of jumping libraries, a suitable enzyme can be used. The enzymes used for DNA fragmentation are very rare, so the fragments obtained are very large. The larger fragments of DNA are then circularized so as to bring the genes that are far apart in close proximity. After the circularization of DNA, these closed sites are again digested with restriction enzymes. The fragments from the closed sites or junctions are cloned together. Such DNA that is cloned from closed site or DNA makes up the jumping library. Chromosome landing. Another technique involved in map-based cloning is chromosome landing. The genes are isolated and identified from a genetic library with the help of this technique. Chromosome walking is tedious and time-consuming to perform if the DNA fragments are extremely large or contain repetitive sequences. The principle behind chromosome landing is at figure 12.41. This technique is highly precise, automated, and robust. However, this technique does not give better results with longer DNA fragments. The dye incorporates and results in unequal shapes and heights of the peak after electrophoresis in the electronic DNA sequence trace chromatogram. Newer technologies have effectively solved all these problems. Are labeled with DDNTPs, which terminate the chains when incorporated in the chains while amplification. Earlier, 4 different reactions needed to be carried out for chain terminator sequencing method, which was tedious and time-consuming. In dye terminator reactions, 4 different dyes are used to label the nucleotides which emit fluorescence of varying wavelengths. Figure 12.41 schematic diagram summarizing the sequencing of a target gene for application in phylogenetic analysis. Molecular markers. Random amplified polymorphic DNA (RAPD). Single primers of random nucleotides are amplified with the genomic DNA. Polymorphisms are detected by these primers, which function as genetic markers. These polymorphisms can then be used for the construction of a genetic map. Heterozygosity or homozygosity cannot be distinguished because the RAPD markers used are dominant. Figure 12.42. The procedure of RAPD is as follows: 1. DNA extraction. 2. Random primer addition. 3. Polymerase chain reaction. 4. Gel electrophoresis to separate the fragments. 5. Addition of ethylene bromide and analysis of RAPD PCR fragments in UV light in order for the results to be reproducible. Strict and consistent reaction conditions need to be maintained. The technique does not require any probe or primer, hence is extremely popular. There is no hybridization or blind steps required as well, which makes the technique efficient, fast, and simple. Figure 12.42. The principle behind the RAPD PCR technique. Annealing sites of the primers are indicated by the arrows. Amplified fragment length. Polymorphism AFLP. Selective amplification of restriction fragments isolated from a complete digest of genomic DNA by PCR is the basis of AFLP technique. Figure 12.43. Steps involved in AFLPR. 1. DNA extraction. 2. Digestion of DNA by restriction endonucleases. 3. Ligating with the adapters. 4. Pre-PCR amplifies the restriction fragments. 5. Selective PCR with labeled primer pair. 6. Gel electrophoresis and analysis of fragments by automated sequencing machines. Programs such as GenMapper can be used to analyze the electrophorograms. This technique has better reproducibility as compared to RAPD. It requires a higher amount of DNA sample. It is technically more complicated. However, the recent advances in automation and the availability of kits have simplified the process largely. Figure 12.43. A schematic flowchart showing the principle behind the AFLP method. PCR-based approach. Rapid cloning has now become a possibility after the invention of a technique known as polymerase chain reaction (PCR). This technique amplifies a segment of DNA million or even billion folds. This purifies the DNA and also generates a large number of copies of DNA for experimentation purposes. The genome sequences are now available for most organisms. This eliminates the need for the construction of a DNA library and genes can be cloned directly. 2 sets of primers are designed, forward and reverse primers. These primer sequences flank the ends of the desired nucleotide sequences of the gene of interest. The DNA isolated from the genome is then heated. The reaction is catalyzed in vitro. DNA polymerase catalysis is the synthesis of DNA. The polymerase used for PCR is derived from a thermophilic bacterium found in hot springs known as Thermus aquaticus. The polymerase is hence named Taq polymerase. DNA polymerase usually cannot withstand the temperature inside a PCR machine and the reaction would not take place. So, we use Taq DNA polymerase. This polymerase is stable at higher temperatures. In the first few cycles, it is extremely difficult to detect any amplification. It is only after several rounds that the power of PCR can be DNA. The reaction is termed a chain reaction because the template for the next reaction is the product from the previous cycle. A single cycle consists of denaturation, annealing, an extension which lasts for 5 minutes. Figure 12.44. Detected. The amount of DNA doubles in every cycle. After a few rounds of PCR, the product we get is a DNA sequence between the 2 primers. Usually, the PCR runs for about 20 to 30 cycles in a reaction that generates enough amplified. Figure 12.44 representation of steps in PCR. There are 3 steps in a PCR. Denaturation, which occurs at 94 OC for about a minute. Annealing occurs at 54 OC for 45 seconds with the help of forward and reverse primers. This step is followed by extension, which occurs at 72 OC for 2 minutes. PCR takes less time for DNA amplification. It makes cell-free molecular cloning a possibility. The whole procedure can be fully automated. PCR is now widely used to clone the DNA directly from a gene of interest. The DNA can be obtained either from the genomic DNA or the mRNA, which is isolated from the cells. PCR method is also awfully sensitive. It is capable of detecting even one DNA molecule which is present in a sample. RNA molecules can also be examined in a similar fashion. They are reverse transcribed to form a cDNA copy which can be examined further. Southern blotting has been replaced widely by PCR due to its ability to detect even trace amounts. The viral infections can now be detected at a very early stage when the viruses are in very low concentration. Forensic medicine is another area where PCR has found an application. Traces of blood and other tissues can be engineered that can be considered a form of super domestication. In the present era, marker-assisted breeding, biotechnology, and genomics are slowly taking over to ensure enhanced crop productivity. Genetic improvement of crop phenotypes by recombinant DNA technology promises a lot of potential to meet the challenges of the 21st century and also offers hope to overcome the stagnation in crop yield as well as produce elite combinations unachievable by conventional plant breeding, the two in a much shorter duration. In 1984, the first transgenic tobacco plant was created, and since then transgenics of many economically important plants resistant to abiotic stress, herbicides, insects, and diseases, and also with superior nutritional and post-harvest quality have been developed. Plant genetic manipulation involves the introduction of gene of interest, termed as transgene, into the vegetative or germline cells of the host genome from unrelated plants or completely different species and its stable inheritance across generations. This horizontal gene transfer has played a crucial role in crop evolution and generated plants with traits that may be entirely lacking in the gene pool of that species. A gene cassette composed of the gene of interest along with correct regulatory sequences, i.e., promoters, enhancers, and terminators are designed and constructed into a suitable vector followed by optimization of protocols for transfer into the plant tissue. It also contains a selectable marker attached to the GOY allowing for selection of the transformed tissue. Gene transfer can be carried out in different ways that are greatly dependent on the plant genotype. Most prefer a particle bombardment and agrobacterium-mediated transformation. Transgene presence in the tissue is screened and confirmed, tissue selected, and then regeneration protocol optimized. Totipotency proves to be a great asset in biotechnology. Once a transgene is stably integrated, it is present in every single cell of the plant. However, its expression is driven and controlled by the promoter. Rate of success in recovering a genetically stable transformant after gene. Detected and examined even if there is a single cell present. The genetic fingerprint obtained can then successfully identify the criminal or the person from whom the cells are derived. Gene transfer in plants. Plants are the reason for the existence of life on Earth. They have been associated with mankind since time immemorial. Although humans emerged long ago, agriculture, which is the most important source of food, has been relatively a recent invention. It has served as one of the bases of modern human civilization and has always been dynamic from the start, both in practice and productivity. Crop breeding is the science of improving agricultural crops in terms of food, fiber, or forage. It deals with methodologies for the formation, selection, and fixation of superior crop phenotypes to suit producer and consumer needs. This artificial selection and genetic modification of wild species to develop a new form of plant more apt for human use is termed as crop domestication. Analyses reveal that the increase in crop production obtained over the years can be attributed to both plant breeding as well as agronomy, each dependent on one another. But plant breeding is a time-consuming process and currently, it alone cannot meet the demands of the burgeoning population. The various biotic and abiotic stresses have also stagnated the yield potential of many crops. With an exponential increase in ever-expanding population, global warming leading to the enhanced occurrence of crop failures, limited availability of land and water, and deteriorating soil profiles, global food security in combination to increased dietary expectations becomes a prime concern. Conventional method of crop breeding has definitely shouldered a lot of this load, and sustainable agriculture methods for increasing crop production like decreasing the use of fertilizer, water, fossil fuels, or other harmful environmental inputs have had important roles to play. With advancements in crop research, modern genetics continues to improve and has been slowly transforming plant breeding into crop replication, and expression is performed by vector. It contains an origin of replication, recombination or multiplying site (MCS), and selectable marker gene. Expression of GOI is under the control of regulatory elements in the non-coding regions of the gene like promoter, enhancer, double enhancer, and silencer. Hence, the two important DNA segments needed for transgenic production of promoter fused upstream of GOI either constitutive or tissue developmental stage specific, ensuring correct expression of trait in the transgenic, and marker gene for selecting transgenics and monitoring the transgene expression. Figure 12.45. Transgene integration can also be verified with polymerase chain reaction, tissue PCR, southern and northern blotting techniques, wherein southern analysis confirms the transgene presence in its copy number, while the presence of transcripts of the introduced gene is assured with northern blotting. Western blotting and immunological methods also help in screening the gene expression, especially in cases where the GOI does not have any enzymatic activity. Integration can be attributed to many factors like the plant genotype, target tissue vegetative or germline, method of stable gene delivery, and screening parameters for monitoring transgene expression. Transformation efficiency is also influenced by phenomena like gene silencing. In the early 1980s, the era of biotechnology began with the breakthrough reports of transgenic production using agrobacterium. By 1996, genetically modified GM crops of canola, cotton, maize, and soybean were commercially available. Since then, this GM technology has revolutionized agriculture, and the area under GM crop cultivation has increased manifolds. The last 2 decades have witnessed a boom in the development of new and refinement of existing protocols. In this section, the methods of gene transfer, screening and monitoring expression of the transgenes and ways to develop marker-free transgenics have been discussed. Marker gene. During transgenic production, the role of transfer of the GOI into the host target for. Figure 12.45 gene cassette. A. The figure shows the original tDNA region which contains genes for auxin, cytokinin, and opine synthesis, and is flanked by the left border LB and right border RB. B. In modified tDNA, this portion in between LB and RB is replaced by transgene or the gene of interest GOI. C. This transgene cassette is actually a combination of selectable marker gene and codon optimized GOI each having its own promoter and terminator sequences. And marker is definitely more than when they are in separate vectors. Even the marker gene expression is regulated by constitutive markers like CAMB, cauliflower mosaic virus, 35S, actin or ubiquitin gene promoters. There are 2 categories of marker genes: selectable markers and non-selectable or sortable markers or reporter genes. Table 12.7. The marker gene thus determines the successful insertion of the transgene into the target. It can be introduced into the plant genome both as part of the same vector as the GOI or on separate vectors, serving as an early and direct indicator for the introduction of GOI. If they are physically linked in the same DNA vector, the frequency of co-transformation, i.e., cells having both GOI. Marker. Types category of marker gene examples of. Marker gene selective agent. SELECTABLE. M. ARKERGENE. Conditional substrate dependent positive NPTII, neo, aficanamycin antibiotic. HPT, HPH, afihygromycin antibiotic. Barphosphenoprison herbicide. Monomanose non-toxic. Negative codified fluorocytosine antifungal. Both positive and negative DAO1D amino acid oxidase non-toxic. Non-conditional substrate. Independent positive IPT non-negative barnase Na. Reporter gene. Conditional WIDA, Gusamug, XLUG non-conditional GFP Na. Table 12.7 categories of marker genes used in plants. Selectable markers. For recovery of transgenic tissue, a selectable marker gene serves as a key component conferring a selective advantage to the transformed cells. It screens early for the putatively transformed tissue followed by regeneration of tissue during which the dying non-transformed cells should have minimal influence on the transformed cells on a selective medium and ultimately helps in producing transgenic plants. They are dominant genes. If placed in the same vector as GOI, it also predicts the number of segregating insertion events that may have taken place in transgenic line. They are taken from both plant and non-plant species, mostly bacterial in origin, E. coli being a major contributor, introducing novel resistance in the target species. But due to differences in the signaling pathways and codon usage between prokaryotes and eukaryotes, the sequences are extensively cells. E.g., transgenic cells with PMI can utilize mannose as a sole food source. Thus, marker genes confer intolerance to growth-inhibiting hormones and salts and antimetabolites are also gaining popularity. In broad terms, a selectable marker gene confers resistance against a selective agent, i.e., shields an organism from a selective agent that would otherwise kill it or not allow its growth. For example, if the vector has an antibiotic marker, it confers antibiotic resistance and will allow growth of only the transformed tissue in the selection medium, minimal medium with a selective antibiotic. That means only the plant carrying the transgene will survive and thus help in the screening of transgenics. It is quite advantageous as the transformation efficiency is generally very low. 1 in several million cells may take up the foreign DNA and it is practically very tedious to check each and every cell. The selective agent will kill all cells without foreign DNA. The selectable markers confer to be of 2 types, either positive promote or negative suppress selection type. Positive selectable marker gene may promote tissue growth and give a selective growth advantage to the cell having the foreign gene. The transformed cells would outgrow the non-transformed ones. Former survive by the action of detoxification or resistance mechanisms encoded by marker, while latter are killed by the use of toxic agents supplemented in the growth media, e.g., NPTII, HPT, BAR, etc. Whereas negative selectable marker genes suppress the growth and selectively kill the transgenic cells. Table 12.8. E.g., CODA, OX2, TMS2, WA, CYP105A, and Q are conditional negative markers. Modified so that they can be recognized by the plant transcriptional and translational machinery forming chimeric genes created to efficiently work in plant cells. Within a plant, the introduced marker gene in no way should interact with specific targets or interfere with any other signaling pathway. That may alter phenotype more than that produced due to transgene or influence any factor, making it difficult to be identified or screened. The first ones designed for plants were those conferring antibiotic or herbicide resistance, former being the most common selective agent. The mechanism of action of the toxic antibiotics in plants is generally inhibition of protein synthesis machinery, particularly in organelle like chloroplast. They normally target metabolic processes and thus prove lethal for the plant cells. Before transformation, plant cell or tissue are grown on a synthetic growth medium. To confirm positive transformance, the growth media is supplemented with the antibiotic. Only those plant cells that contain the transfer-resistant gene can grow in them. Neomycin phosphotransferase 2 gene NPTII is the foremost and one of the most exploited in plant biotechnology conferring resistance to kanamycin. Along with NPTII, those having HPT and BAR genes conferring hygromycin or vascular resistance, respectively, have been used for more than 95% of the GM reports. Genes that are dependent on non-toxic agents have also been identified like mannose-dependent selectable marker gene or phosphonose isomerase gene (PMI) and proved to be an alternative to the toxic chemicals like antibiotics or herbicides. Those markers only suppress the growth of non-transformed genes, enzymes, substrates, use of substrate. Codocytosine deaminase. 5-fluorocytosine antifungal medication. OX2 aminohydrolase naphthalene synthetic auxin. TMS2 indole acetic acid hydrolase. Indole 3-acetamide fungal metabolite, a bacterial metabolite in a plant. Metabolite. DWA dehalogenase dihalocanes bioremediation, decontamination, biosensing of. The positive selectable ones are preferred in plant biotechnology. Out of them also, conditional positive selectable marker genes are the most important ones preferred for recovery of transgenics, i.e., can be conditional on external substrates or have the ability to detoxify a substrate in the growth media. The table 12.9 lists some selectable marker genes used for conditional positive selection during plant transformation. Non-conditional ones do not use any external substrate but alter the growth and development of the transformed cells. Although non-conditional negative markers have not been much used for GM crop production, but have been used in crop breeding, they may cause cell death of specific cells. E.g., ribonuclease barnase extracted from Bacillus myelocalications. S number, selection. Marker gene. Selective agent, antibiotic. Herbicide, drugs or analogs, enzyme encoded. 1. Antibiotics toxic. Neo NPTII, APA2. Kanamycin and structurally related neomycin, paromomycin, ribostomycin, butyrosin. Gentamicin B, and geneticin G418. Aminoglycoside. 3-peat phosphotransferase 2 APH, 3-peat IIA, or neomycin phosphotransferase 2S number. Selection. Marker gene. Selective agent, antibiotic. Herbicide, drugs or analogs, enzyme encoded. VNPTI, alpha-1 G418 neomycin phosphotransperases. CHPT, HPH, FIB. Hydromycin B, hydromycin B phosphotransperases. DSPT, streptomycin.